Good day, Doctor. My name is Nurul Binti Shamsul, Nizam, now measuring at 79004. Today, I will be speaking about pilocarpine, a direct-acting cholinergic agonist. Primary clinical application focus on the palliative management of dry mouth or xerostomia resulting from head and neck radiotherapy or Sjogren's syndrome. It is typically administered 3 to 4 times daily based on patient tolerance, while historically used for ophthalmic condition like dry eye or glaucoma. Modern practice largely favor T-cell modulators like cyclosporine and tacrolimus, leaving pilocarpine core role as a dose-directed treatment for exocrine gland stimulation. On this slide, you can see visual example of pilocarpine formulation and related medical contents. Sjogren's syndrome, the infographic on the left, highlights Sjogren's syndrome, an autoimmune condition targeting tear and salivary glands, leading to severe dryness. And for ophthalmic solution, the image on the right showcases commercial preparations such as pilocarpine, hydrochloride ophthalmic solution traditionally used in glaucoma management to lower intraocular pressure.
Assalamualaikum warahmatullahi wabarakatuh. My name is Amirah Zahra Binti Zamri, Nometry as 81256. Today I would like to present about pilocarpine, focusing on its application and chemical structure. Let's begin with its application. Pilocarpine is a naturally occurring alkaloid that acts as a muscarinic acetylcholine receptor agonist. In simple terms, it mimics the action of the acetylcholine and stimulates muscarinic receptor in the body. One of the main applications is in treatment of glaucoma and ocular hypertension. When pilocarpine is applied to the eye, it stimulates the iris, sphincter, and ciliary muscle. This causes miosis or constrictions of the pupil and help increase the drainage of the aqueous humor. As a result, the extraocular pressure is reduced, which is important in preventing further damage on the optic nerve. The second important application is the treatment of xerostomia or dry mouth. Pilocarpine stimulates the muscarinic receptor in the salivary gland, which increases saliva productions. Therefore, it can help patients with conditions such as Sjogren's syndrome and patients who experience dry mouth following radiotherapy or head and neck cancer. Increased saliva can improve important daily activities such as eating and speaking. Finally, pilocarpine can also be used in symptom ophthalmic preparation for presbyopia, which is an age-related condition that affects our ability to focus on nearby objects. By stimulating the ciliary muscle and causing miosis, pilocarpine can temporarily improve near vision. So overall, the main therapeutic effects of pilocarpine are related to its ability to stimulate muscarinic receptor, particularly in the eyes and salivary glands. Next, I will explain the chemical structure of pilocarpine. Pilocarpine has a molecular formula C11, H19, and 2O2, which a molecular mass of approximately 208.26 grams per mole. Structurally, pilocarpine contains two important heterocyclic components. The first is a gamma-butyrolactone or lactone ring, which contains a cyclic ester functional group. The second is an N-methylamidazole ring, which contains two nitrogen atoms. These structural features are important because they contribute to the chemical properties and biological activity of pilocarpine. Another important feature is the stereochemistry. Pilocarpine contains two stereogenic carbon atoms, meaning that the three-dimensional arrangement of its atoms is important for how the molecule interacts with its biological target. The naturally occurring from is plus-minus pilocarpine, which have a specific three-dimensional configuration. The opaque name of pilocarpine is shown on the slide. Therefore, although pilocarpine is a relatively small molecule, its functional group, heterocyclic rings, and stereochemistry all contribute to its biological activity. That's all for me. Thank you.
Moving on to the classification of pilocarpine, it can be classified two main ways, chemically and pharmacologically. From a chemical perspective, pilocarpine is classified as a naturally occurring imidazole alkaloid. Alkaloids are naturally occurring nitrogen-containing compounds that often have significant biological activity. Pilocarpine is obtained naturally from plants belonging to the Pilocarpus genus, particularly Pilocarpus microphyllus, which is commonly associated with the plant known as jaborandi. The term imidazole alkaloid refers to the presence of imidazole rings containing two nitrogen atoms within its structure. From a pharmacological perspective, pilocarpine is classified as a direct-acting muscarinic cholinergic agonist. It is also considered a parasympathomimetic drug because its effect resembles those produced by activation of the parasympathetic nervous system. By directly stimulating muscarinic acetylcholine receptor, pilocarpine produces effects such as increased salivation and contraction of certain ocular muscles. So, to summarize, pilocarpine is an imidazole alkaloid from a chemical perspective, while pharmacologically it's a direct-acting muscarinic cholinergic agonist.
Okay, so for my part, I will explain the purpose of building block involved in the biosynthesis of the pilocarpine. Based on the available evidence, three important compounds have been proposed to be involved in the formations of pilocarpine, such as L-histamine, L-theanine, and S-adenosine L-methionine, or we can call it SAM. So L-histamine and L-theanine are proposed as a precursor that contribute to different parts of the pilocarpine structure, while SAM is proposed to act as a methyl donor during the final stage of the biosynthesis. It is important to note that this pathway is still considered proposed because the complete biosynthetic pathway of pilocarpine has not been experimentally confirmed. So first, L-histamine is proposed to contribute to the imidazole moiety of the pilocarpine. It can be converted into the urocanic acid by histone ammonia lysis, or we can call it HAL. Next, L-theanine is proposed to contribute to the carbon skeleton. It can be converted into the alpha-ketobutyrate through the action of the theanine ammonia lysis, or we can call it TAL. Finally, SAM acts mainly as a methyl donor. It is proposed to provide the methyl group during the N-methylation of pilocarpine to form pilocarpine. So in the simple terms, L-histamine contribute to the imidazole part, L-theanine contribute to the carbon skeleton, and the SAM provide the methyl group. However, the complete pathway is still proposed and has not been fully confirmed experimentally. Okay, so next I will explain the biosynthesis pathway. And these are the diagram of the biosynthesis pathway. We can have a look. So moving on to the purpose of biosynthesis pathway of the pilocarpine, basically the pathway is proposed to start from two precursor molecules such as L-histamine and L-theanine. L-histamine forms one branch of the pathway and is mainly associated with the imidazole containing part, while L-theanine forms another branch that contributes to the carbon skeleton. These two branches then undergo several proposed transformations and eventually come together to form a common intermediate compound 2. From the compound 2, further structural change leave compound 1, which is 3-nol-7-11, and dihydropilocarpine, which is then proposed to undergo reduction to finally form the pilocarpine. So in a simple way, we can think of the pathway as two precursor pathways coming together, followed by several modifications to finally produce pilocarpine. However, some of the steps are still proposed and the enzyme involved had not been fully identified. So this pathway is not yet completely confirmed.
Moving on to the synthetic chemistry aspect, this slide outlines the 7-step laboratory pathway to obtain pilocarpine starting from 2-ethyl 3-carboxy 2-butyrolactone. For the step 1, the starting carboxylic acid is converted to an acid chloride using thionyl chloride SOCl2. For the step 2, we perform an ester homologation using diazomethane CH2N2 followed by ethanol treatment to form an ethyl ester. For the step 3, acid hydrolysis regenerates the carboxylic acid, which is again converted to an acid chloride with SOCl2. For the step 4, treatment with H2N2 followed by HCl yield a chloroketone. For the step 5, reaction with potassium pentanimide and subsequent acidic hydrolysis yield the aminoketone intermediate. For the step 6, condensation with potassium thiocyanate KACN under acidic condition construct the merceptomidazole lactone core. For the last step, finally, oxidation using concentrated nitric acid and sodium nitrite deliver pilocarpine. The initial yield was 84%, which was successfully optimized to 92%. And ultimately, pilocarpine hydrochloride was isolated in 79% yield with a melting point of 200 under 203 Celsius, showing spectroscopic data consistent with authentic material. This slide illustrates the step-by-step chemical structure corresponding to the pathway just described, beginning with lactone intermediate, 13.1.15 acid chloride formation leads into homologated carboxylic acid, 13.1.16. Conversion to chloroketone 13.1.18 allow for Gabriel-type glycophilic substitution, yielding aminoketone 13.1.19. Imidazole ring closure via thiocyanate addition provide intermediate 13.1.20 followed by oxidative desulfurization to yield the imidazole derivative 13.1.21. Methylation with methyl iodide CH3I yield compound 13.1.22, completing the skeletal framework of pilocarpine.
For the conclusion, pilocarpine is a naturally occurring imidazole alkaloid with important pharmaceutical application, especially in treating glaucoma and dry mouth. Its biosynthesis is proposed to involve L-histidine, L-threonine, and SAM, although the complete pathway is not yet fully understood. Pilocarpine can also be synthesized in the laboratory through several chemical reactions, showing its importance in natural product chemistry and pharmaceutical development.